Welcome to the EPG Pathshala. I am Dr. Shivani Sharda, working as Assistant Professor at Amity Institute of Biotechnology, Amity University, Noida. Here in this module, we are talking about transfer RNA and its processing events. Transfer RNAs are indispensable entities for protein synthesis. Their biosynthesis recruits specialized RNA polymerases that effectuate preliminary transcripts which are eventually transformed via a host of post-transcriptional refinements prior to tRNAs turning as mature molecules for their commission role in protein synthesis. The predominant disposition of transfer RNAs to act as the salient molecule, decoding the genetic instructions held on messenger RNA with a vital capacity of furnishing amino acids explicit to the mRNA codons for the cytoplasmic and organellar protein synthesis machineries. This adopter function of tRNA necessitates it to perpetuate the canonical L profile structure, which is crucial to its decrypting function. The cytoplasmic transfer RNAs transcribed in the nucleus undergoes immense alterations, and these processes contributes to consign processed or partially processed tRNAs to accurate subcellular location, tRNA trafficking, RNA refolding through enzymes or RNA chaperones, as well as the degradation of misfolded tRNAs and tRNA quality control. The processing events associated with tRNA synthesis and maturation serve errands encompassing creation of tRNA, discriminant pool, translation fidelity, and subcellular dynamics, implicating their importance for the functional molecular controls. The content throws light on the events associated with tRNA processing and modification to generate functional tRNA molecules through genetic, genomic, biochemical, and cell biology investigations undertaken. The production of tRNA in organisms of prokaryotic, eukaryotic, and archaeal origin requires post-transcriptional processing, and the resultant tRNA has distinctive nucleotides and a specified folding pattern that constitute similar final structure and function. The tRNAs can be produced simultaneously with rRNAs or the ribosomal RNAs, or transcribed as a part of larger pre-tRNA transcript that is cleaved to generate multiple tRNA molecules. Eukaryotic tRNA genes occur in cluster on specific chromosomes, where individual cytoplasmic tRNAs gets transcribed in the nucleus by dedicated transcription through DNA-dependent RNA polymerase III. Thus, a single pre-tRNA is produced from each gene. Bacterial and eukaryotic tRNAs require processing before they assume their functional role of transporting amino acids to the ribosome, wherein a lineage of post-transcriptional modulations and alterations processing into the standard length and modified so as the canonical L-shaped structure required for its adopter function for mRNA coding is maintained. The partially transformed and processed transfer RNAs are relocated to the precise subcellular setting where tRNA trafficking is an elaborate progression. The tRNAs are mostly stable molecules, though they are resident pathways that cater to the degradation of the partially processed or misfiled tRNAs and the quality checkpoints. The content focuses on the modification procedures and post-transcriptional events that are required for the biogenesis, turnover, and intracellular dynamics contributing towards functional tRNA processing. Let us look at the detailed information of the processing events in transfer RNAs. The first event is the synthesis of precursors of tRNA by RNA polymerase III, which is also termed as transcription initiation. The transcriptional initiation process is progressed through various steps as outlined in the slides. The promoter region of tRNA genes possesses two consensus sequences that fall downstream of the transcription start site. These sequences are termed as box A and box B. There have been demarcation of two complex DNA binding factors that are precursors to transcription initiation by RNA polymerase III, termed as transcription factor C and transcription factor B. Transcription factor C, a large protein complex, is the assembly factor for liaising transcription factor 3B at the correct location, while the latter is the literal initiation factor for RNA polymerase III. Transcription factor 3B is characterized as three subunits entity containing of TBP, B and BRF domains. The molecular events associated with the transcriptional initiation are transcription factor 3 binding leading to the transcriptional start sites at box A and box B of the tRNA promoter. Transcription factor 3b binding to transcription factor 3c DNA complex 50 base pair upstream from transcription factor 3c binding site at box A, then followed by RNA polymerase III binding. The transcription factor 3b furthers the RNA polymerase III recruitment that initiates transcription, probably repositioning the transcription factor 3c from DNA template while moving onto the strand. The transcriptional termination is mostly achieved without adjunct factors through the DA cluster residues, with the overall efficiency depending upon the adjacent sequences. The steps that are broadly involved in transfer RNA processing are briefly delineated as transfer RNA transcription by RNA polymerase. The transcription product, the pre-tRNA molecule, contains additional RNA sequences at both the 5' and 3' ends. These additional sequences are removed from the transcript during processing. The additional nucleotides at the 5' end are removed by an unusual RNA containing enzyme called ribonuclease P, or abbreviated as RNase P. Furthering into the details of the pre-tRNA processing includes the following events schematically depicted in the slides. The first one being the cleavage of the 5' leader sequence. The wiping out of the 5' leader sequences from the initial tRNA transcript prevails before effectuating the 3' extensions. Both prokaryotic and eukaryotic tRNAs contain extra nucleotides at the 5' end that are severed by endonuclease RNase P. This enzyme is a ribozyme comprising a catalytically active RNA molecule and small basic protein subunits capable of catalyzing chemical reactions on its own, also termed as ribonucleoproteins. The second event is the removal of the 3' trailer residues. The eradication of the 3' extensions from the pre-tRNA is an intricate process that necessitates inclusion of both exo and endonucleases that advocate competitive differential access to tRNAs for end maturation. The secondary structure stems and loops possessing the extra nucleotides at the 3' end, that is present in both prokaryotes and eukaryotes, are cleaved by endonuclease RNase E or F at the middle of the 3' trailer sequence or at the base of the stem, leaving some extra nucleotides. These extra nucleotides are snipped through exonucleotic reduction of the surplus trailer sequence by RNase D, RNase PH from the 3' end in a one-step manner. The further processing event involves the addition of trinucleotide CCA at their 3' end. established by the template-independent RNA polymerization by the CCA adding enzyme. All the tRNAs include a small 3-terminal CCA sequence vital for tRNA aminoacylation and interaction with the ribosomes, exhibited vividly in the protein biosynthesis. The CCA sequence is synthesized de novo or reconstructed by a template-independent RNA polymerase, that is the CCA adding enzyme, utilizing CTP and ATP as energy substrate. These three bases are not coded for by the tRNA gene. Instead, these nucleotides are added during processing of the pre-tRNA transcript. The enzyme responsible for the addition of the CCA end is tRNA nucleotidyl transferase, and the reaction proceeds with utilization of the molecular energy. The CCA adding enzyme is a unique nucleotidyl transferase that stretches out a specific primer, that is tRNA, by annexing an explicit sequence CCA, as well as it supervises the tRNA 3' terminus status and reconstructs if needed. Whereas the prokaryotic tRNAs are encoded with the CCA sequences, that is, they possess a gene encoding the CCA adding enzyme, the eukaryotic nuclear encoded tRNA 3' CCA sequences are formed definitely through post-transcriptional nucleotidyl transferase catalysis. The next step is the tRNA splicing or removal of introns from tRNA precursors located in the anticodon arm. This step in the tRNA processing is mostly limited to some eukaryotic pre-tRNAs and archaeal pre-tRNAs that possess introns subsequently removed by the endonucleatic cleavage of both intron ends, accompanied by the ligation of the half molecules of tRNAs. The tRNA introns are consistently present one base 3' to the anticodon, making the extrication of introns by tRNA splicing a requisite process, so as to prompt a complete set of tRNAs meant for decoding. The function of tRNA introns is implicated for nucleoside modifications in the anticodon loop. The pre-tRNA splicing reaction is mediated by an endonuclease, that is a heterotetramer that results in 2 tRNA half molecules after removal of introns, which are then ligated by the 3' to 5' activity. The fourth type of modification is the base and ribose unit modification through the enzymatic changes of the standard ribonucleotide in a tRNA precursor, resulting in unusual bases. Mature transfer RNAs can contain up to 10% bases other than the usual adenine, guanine, cytidine and uracil. tRNAs are efficiently modified with a plenitude of known tRNA nucleoside refinements. The modification here comprehends methylation, acetylation, deamination, reduction, rearrangement, attachment of isopentenyl or SH groups of bases. These alterations, occurring approximately in 1 in 10 nucleotides identified in tRNAs, conjure the diversity permitting significant structural and functional flexibility. The tRNA modifications as described achieve manifold functions, specifically targeting tRNA discrimination, maintaining translational fidelity through selective codon-anticodon interaction, sustenance of mRNA reading frame and tRNA stability. The events in tRNA maturation are elaborated as post-transcriptional processing of tRNA, where processing of tRNA synthesized as precursor include cleavage by nucleases and other modification of the nascent RNA chains. The tRNA molecules are either snipped out from a single primary RNA transcript or from transcripts that embrace arrays of scattered tRNAs or multiple copies of the same tRNA. The tRNA maturation steps are connoted as initial transcription of tRNA by RNA polymerase III as precursor molecules termed as pre-tRNA with 5' and 3' extension sequences called the leader and trailer sequences respectively. The two sequences which are extensions at the 5' and 3' end are cleaved by azotid enzymes so as to snip the precursor to the desired standard length. This event is followed by modifications through modification enzymes that include incorporation in the tRNA core, landscape bordering of the D and the T loop as well as the anticodon loop of the unprocessed transcript, intron removal, discrete nucleoside modifications, for example, sugar methylations, base deamination, base isomerization and addition of exotic moieties to the bases, resulting in a mature transfer RNA. tRNA processing and regulation is demarcated through its turnover, cleavages and subcellular trafficking. Transfer RNAs have stable half-lives and follow separate pathways for tRNA turnover that are bound to function in tRNA quality control, eliminating inappropriately processed, modified or folded transfer RNAs from the generated pool of transfer RNAs. The overall impact of tRNA modifications ensues the accurate decoding processes, ensuring appropriate ribosome interaction, tRNA discrimination by cognate amino-acyl tRNA synthetases and its stability affecting the translational programming. The continued studies on the processing of transfer RNAs throw novel information into the protein RNA specificity and the structural pathways of metabolically linked molecular coordinations. In the present scenario, these are the best documented events and associations of tRNA processing activities that become a part of transfer RNA biogenesis, where the investigations of tRNA alterations and the stability gives the due worth of the importance in cellular metabolic processes. tRNA biology demarcates the essential roles of transfer RNA processing and regulation at its biosynthesis that embrace newer molecular discoveries and surprises to come. The details of such events are described in the e-text linked to the module. You are advised to go through the associated references for further details. Thanks for watching the details on the molecular steps analogous to transfer RNA processing. For the messenger RNA and ribosomal RNA processing, please visit the respective modules and learning materials. The adjunct attributes of the processing and regulation modules are in the write-up provided in the module. The references can be consulted for detailed information of particular examples. The quiz linked to the e-text further strengthens your information on the module. Please visit the EPG Pathshala for the related biology topics.